Hello everybody, Dr. Lloyd here. This is chapter 3, Cells, the Living Unit. Just want to warn you, this chapter is very long. And let's get through it. Okay, so the aim of the chapter is to introduce cells, cell parts, membrane transport, cell division, protein synthesis. So those are the three topics. So first we'll start out with what's called cell theory. So a cell is a structure and functional unit of life. There are over 250 different types of human cells, different size, different shape, different subcellular components, and these differences lead to different function. So here we've got just a few different cells, and human cells have basically three parts: the plasma membrane, which is the outer flexible covering, the cytoplasm, which is the intracellular fluid, kind of like a jelly, which contains all the organelles, and the nucleus, which contains the DNA and is the control center. So here we have all of the organelles. It's a good idea to put each one of these on a flashcard and just put one or two functions. So extracellular, the extracellular fluid bathes the cells, and this would be blood plasma, which would be the fluid in blood. It could be cerebrospinal fluid, interstitial fluid, which is the fluid between cells. You have other fluids like secretions, saliva, and mucus. So let's start with the cell membrane. So it acts as an active barrier separating the intracellular and extracellular fluid. Plays a dynamic role in cellular activity by controlling what enters and what leaves the cell. It's kind of like the gatekeeper. So the cell membrane consists of membrane lipids that form a flexible lipid bilayer, and we're going to look at that structure in just a second. It's also been called the fluid mosaic model, looking at the fluidity of the membrane and all of its components. There are surface sugars called the glycocalyx, as well as cell junctions that hold the cell together. Here's a picture showing the lipid bilayer. So bi means two, so there's two layers to this cell membrane. You've got the top layer and the bottom layer, and I'll explain why it's arranged like this. And we have different proteins. And on the extracellular compartment, you can see these hexagons, which are basically glucose chains. So this is part of your glycocalyx. Okay, so if we look at the constitution of the membrane, you've got your phospholipid, which has a polar head group, which is hydrophilic, and you've got a nonpolar tail, two tails, and that is hydrophobic. Okay, so half of it likes water, half of it fears water. And then we got cholesterol. Cholesterol affects the fluidity of the membrane. We've got proteins, integral, transmembrane, as well as peripheral proteins. We'll get to those. And then the carbohydrates, part of the glycocalyx, which we just covered. So the lipid bilayer is made mostly up of phospholipids, and it's got a phosphate head and a fatty acid tail. And so here's a picture of it. So this head is charged. It's got negative charges on it, and therefore it will hydrate, so you'll get water around it. The tails, however, will push out the water, and they will sandwich in with another tail and another tail to make a water-free environment. So with this lipid bilayer, you can see there are a couple different types of proteins. There's the integral transmembrane proteins that go all the way across the membrane. Okay. Then there are peripheral proteins that just sit on the periphery. Sometimes they're anchored to the transmembrane proteins, sometimes they're not. So the integral proteins also have hydrophobic and hydrophilic regions. The hydrophobic regions, remember, water-fearing, those interact with the lipid tails, while the hydrophilic interact with the water components, whether it's extracellular or intracellular. And so the peripheral proteins are loosely attached, and they can function as enzymes, as motor proteins, as cell-cell connections. So many tasks of the membrane. The first one is transport. So there's a channel that's formed through the transmembrane proteins, these integral membrane proteins, and it's a hydrophilic channel because it allows water, and it's selective for a particular solute. Some use ATP, some do not. Receptors. So when bound, the chemical messengers cause a shape change in the protein. It's a conformational change that will initiate some chain of reactions leading to some kind of signaling. Enzymatic activity. Some integral membrane proteins work as enzymes with active sites, and you can see them processing substrates here. Cell-cell recognition. Well, we talked about the glycocalyx and the glycoproteins. These are involved in the recognition of cells. Attachment of extracellular matrix. A lot of matrix. When I mentioned that the cytosol is jelly, it just means that it's water with a lot of cytoskeleton that kind of makes it a little bit thicker. Cell-cell joining. There are different cell adhesion molecules that combine cells. And cell junctions. Let's see. So there are desmosomes. Desmosomes are they interlock cells like a zipper. Okay, desmosomes do allow some kind of give between cells. Therefore, cells can move without tearing. Another cell junction is the gap junction. These are made of proteins called connexins. They form tunnels and allow molecules to pass. These are usually small ion simple sugars. So the plasma membrane is selectively permeable, allowing only certain substances to cross. There's passive transport that requires no energy, and there's active transport, which requires the hydrolysis of ATP. So there are three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. So diffusion is simply the movement of molecules from an area of high concentration to an area of low concentration. This is the concentration gradient. So the greater the concentration difference, the faster the diffusion. The smaller the molecule, the faster the diffusion. The higher the temperature, the faster the diffusion. And so here we show a dye pellet being put at the bottom of this water beaker, and over time it becomes all one color, it becomes all blue even. That's from the molecules bouncing around and mixing all by themselves. They don't need you to mix it, they will mix themselves. Okay, simple diffusion, mainly nonpolar lipid soluble substances that go directly through. Okay, these are going to be like oxygen, okay, carbon dioxide, your two gases that are very important for cellular respiration. Then there's facilitated diffusion. Facilitated really means helped. Okay, so it's diffusion, but down a protein, down an integral membrane protein. So it allows things like glucose and amino acids and ions to passively move down the gradient. Then there's carrier-mediated facilitated diffusion. So this is transmembrane integral proteins transporting specific molecules. So here we've got diffusion. We see conformational changes in the protein, and it grabs onto the specific solute and it pumps it inside. And it looks like no ATP is required for this reaction. So there are also channels that mediate diffusion. It's called channel-mediated facilitated diffusion. And an example would be aquaporins. These aquaporins let water through. Remember, water is polar, so it doesn't readily go through the membrane, but it goes down these aquaporins. So osmosis is a movement of solvents such as water across a selectively permeable membrane from an area of high concentration to an area of low concentration of water. Okay, and osmosis is going to occur through aquaporins. Okay, tonicity. So there are three different types of solutions when you have outside of the cell and the inside of the cell in like a beaker. Okay, an isotonic solution would be the same salt sugar concentration on the outside and the inside, so the cell volume remains unchanged. If it's a hypertonic solution, it means that you have more ions outside of the cell, and therefore the cell shrinks. Okay, it's trying to balance the contents by having diffusion of the substance from a high concentration to low concentration. Water follows. Hypotonic solution would be the opposite, where you have a higher salt sugar concentration on the inside of the cell, and the cell will swell. So this is just a picture of that. So isotonic, you see these red blood cells. Red blood cells are very susceptible to differences in tonicity. Isotonic is when you have no net movement in and out of the cell. The red blood cell looks like a biconcave disc, and it keeps its shape. Hypertonic, this is when there's more salt on the outside, and osmosis is going to occur to try to even out the water concentration, and water is going to leave the cell. The cell is then going to shrivel. This can be devastating for the cell. Then the opposite, when you've got a hypotonic solution, it means that the outside has less ions and sugar than the inside. And so to balance that out, water is going to move inside the cell, and this will make the cell very plump and will lyse the cell eventually. So a good example would be like if you're giving somebody intravenous saline, and you mistakenly give them intravenous water without the salt, without the saline. This is going to lead to widespread bursting of red blood cells, because that's a hypotonic solution, and that can be deadly. So there's active transport and vesicular transport, both of which require ATP. Some transporters are antiporters, means they put one solute one way and the other solute the other way. Some are symporters, where you have two different substances going the same direction. And these antiporters and symporters require ATP. There are different types of active transport. So when we say active transport, we say is ATP dependent. It's going to use ATP. So there's one large protein called the sodium-potassium ATPase. This is a very important enzyme that ejects sodium and injects potassium. So naturally, the extracellular fluid is high in sodium. It's like the ocean. The ocean has full of salt water, and that's like extracellular fluid. The intracellular fluid, however, is high in potassium and low on sodium. Now, the potassium can leak out of the cells, and this is what establishes the membrane potential. So the difference in voltage from the outside to the inside. With potassium, it's a positive charge. When it leaves the cells with a net negative charge. Okay, so there is different types of active transport, coupling to certain other pumps. I'm not going to go through that too much. Okay, vesicular transport. This is larger particles. Okay, so we're no longer talking oxygen or CO2. We're talking macromolecules, and this does require ATP. And so you have endocytosis, where the cell is eating, like phagocytosis, or the cell is drinking, like pinocytosis, or it's receptor-mediated endocytosis when its receptor is activated. And then you have exocytosis, and then also transcytosis. So transcytosis would be movement across the cell. So endocytosis involves protein-coated vesicles. There are receptors for that. These are clathrin-coated pits, but they take in what it is on the outside. And specifically, if it's a solid they're taking in, then it's referred to as phagocytosis, or cell eating. If it is liquid they're taking in, it's referred to as pinocytosis, or cell drinking. Exocytosis is when you have an efflux or exiting of cellular components through these vesicles. These vesicles basically just open up and dump the components out. This could be waste, could be storage. Okay, we mentioned a little bit about potassium and the resting membrane potential, and how potassium establishes the resting membrane potential, while the sodium-potassium ATPase maintains the resting membrane potential. So cells range from minus 30 to minus 50 to minus 100 millivolt charges. And so they're negative 50 millivolts with respect to the outside, or negative 100 with respect to the outside. Now, what establishes the membrane potential is the high potassium on the inside and the high sodium on the outside. As long as those two ions are separated, you're going to have a change. You're going to have a membrane potential. If the ions all equilibrate, that's cell death, okay? Where no movement, no work can be done. Okay, so here we can see the potassium on the inside and the sodium on the outside. There's also chloride. Don't worry about chloride. And so we're showing potassium leaking out. That's making the cell more negative. Okay. Because you're losing this positive charge. Okay. So let's go on to the cell, the different components of the cell: the mitochondria, endoplasmic reticulum, Golgi apparatus, peroxisome, lysosome, and then those are membranous. Okay, they're membrane bound. Non-membrane bound are ribosome, cytoskeleton, centrioles. Okay, so the mitochondria are the powerhouse of the cell because they make ATP. This is where cellular respiration occurs. Now, mitochondria, interestingly enough, have their own DNA and their own RNA and their own ribosomes. And we can get into what's called the endosymbiotic theory later. This is how they look. They have this inner membrane and cristae that are wrapped. But this is where oxidative phosphorylation and breakdown of carbohydrates occurs to produce ATP. That's why they're the powerhouse. So the ribosomes, there are a couple of different kinds. There are free ribosomes or membrane-bound ribosomes. But ribosomes are basically RNA, which we can talk about in a minute. And they're the site of protein synthesis. So they have a little pocket where the different amino acids go into and form a peptide bond. And this is how proteins are made on ribosomes. The endoplasmic reticulum, This is a series of membranes, and it comes in a rough and a smooth. The rough ER is the ER with ribosomes attached, where the smooth ER does not have ribosomes attached. And so we can see here, the rough ER has these dots. Those dots are ribosomes. Okay, so the rough ER is the site of protein synthesis that is secreted from the cell. Okay, so these vesicles are created. And the smooth ER is the site of lipid metabolism, cholesterol and steroid-based hormone synthesis, absorption synthesis, detox, all sorts of different processes. The Golgi is like the FedEx of the cell. It takes these products that was made by the endoplasmic reticulum and it ships them around the cell. So here you see the Golgi. It's receiving vesicles from the rough ER or the ER. They bond with this membrane system. They're processed, sorted, and then released and maybe exocytosed. Okay, peroxisomes are very powerful neutralizing agents. Okay, so peroxisomes have toxins and they break down fatty acids and other species. So they're a strong oxidative component. The lysosome is like the stomach of the cell. It has digestive enzymes, and so whatever is endocytosed, let's say a bacterium is endocytosed, it will then go into a lysosome where it gets digested and breaks the bacterium down into its component parts. The cytoskeleton, okay, it's made of microfilaments, intermediate filaments, and microtubules. And these are very important to maintain cell shape, because these are not plant cells. We don't have a large cell wall. These are cells that can change shape based on your cytoskeleton. So there are microfilaments, which are the thinnest of all the cytoskeleton. These are mainly strands of actin. Here you go. You just a picture of some. Intermediate filaments are a little bit larger, and these are fibrils. So they're tetramers twisted together in one strong fiber. These are very tough. And then you have microtubules, which are the largest. These are hollow areas that have a centrosome. They can interact with motor proteins and act as ropes that can pull certain areas of the cell. The motor proteins can walk on this region. And so here it shows tubulin subunits, and then a cell down here with all the microtubules stained, so you can see all of this cytoskeleton here. So the motor proteins function in motility, and they are powered by ATP. Okay, centrosome. Well, the centrioles, a pair of barrel-shaped microtubules, organelles, that lie at right angles to each other, and these function in cell division. You'll see them pop up and light up during cell division. They also form cilia and flagella. And so here are some centrioles. Here's a nice picture. So cilia and flagella aid in movement of the cell and movement of species across the cell, where microvilli are finger-like projections of the plasma membrane that extend, and this increases surface area, like in the stomach when you want to have to improve absorption. So the cilia whip, they move in a whip-like motion, like in your trachea when you cough something up. That phlegm is coming up the trachea via cilia. Then you also have flagella, like the tail of a sperm. Flagella have a 9-2 pattern. We talked a little bit about microvilli, these finger-like projections that increase surface area. Here we go. So we'll see a lot of these in the digestive tract to increase absorption. Okay, nucleus, the largest organelle. Most cells have one nucleus, but that's a lot of cells have multinucleus as well. Some cells fuse and therefore have a couple nuclei because it's fused with another cell. Now, red blood cells are anucleate, which means they don't have a nucleus. They have lost their nucleus. So the nucleus has an envelope that surrounds it, and then it has a little dot in the middle called a nucleolus. And then in the rest of the nuclei, there's scattered chromatin. This depends on what stage it's in. Okay, there are also nuclear pores that let species in and out. All sorts of nucleic acids transport out of the nucleus. So the nucleoli, the small dot in the nucleus, this is the synthesis of ribosomal RNA. Okay, very important in protein synthesis. Where the chromatin is the relaxed DNA, the chromatin is going to change when we get to the cell cycle. But in a normally growing cell that's not undergoing cell division, the DNA consists of chromatin. So it's just relaxed DNA, kind of looks like sand. Okay, let's get to the cell cycle. So there are different periods. All the time in between the cell cycle is called interphase. That's when you're going to see all of your chromatin. So there are subphases in the cell cycle, and I'll show you a picture of this, but there's a gap 1, a synthetic phase, and a gap 2. So gap 1 and gap 2 are for cell growth, and S phase is for DNA replication or DNA synthesis. Before you divide your cell, you need to double your DNA so that when you divide, each cell gets the same amount of DNA. If you didn't double your DNA, each time you divided, you'd have half your DNA, and then half of that DNA, and half of that DNA, and so eventually you'd wind up with no nucleic acid in your cells. So what happens is, before the cell divides, it replicates, it duplicates its DNA. It's called DNA synthesis. Here's a nice picture. Okay, we've got the cell cycle. Interphase is 90% of the cell cycle. During this phase, you've got growth, G1. You've got the synthesis of DNA, the doubling of DNA. And then you've got G2, which is another growth and final preparation. Then we enter the M phase, which is the mitotic phase. This is mitosis. Mitosis is cell division of non-gametes. Okay? All the rest of your somatic cells, your body cells, are going to undergo mitosis, and just the gametes are going to undergo a special process called meiosis. But there's PMAT in mitosis. P for prophase, M for metaphase, A for anaphase, T for telophase. That's PMAT. And then at the end you have cytokinesis. And so we're going to go through these. You got some checkpoints. So the cell says, are we ready to divide? Okay, that's one checkpoint. The second checkpoint, did we divide? Are we growing properly? So if you fail the checkpoint, chances are the cell is going to commit suicide, okay, and just disintegrate. Okay, so in DNA replication, there's a lot of details. We're going to gloss over these, but there is a replication fork and a replication bubble. DNA polymerase is your enzyme complex that is going to synthesize the DNA. So here we go. We've got our double-stranded double helix that unwinds via an enzyme called helicase and leads to two different strands. These strands are read by DNA polymerase, which then makes a second copy on each strand. So each strand gets a new copy. So you have two old strands and two new strands. It occurs in different processes like just the leading strand and lagging strand. We won't go into that. Replication bubble is this bubble that's created. So mitosis is the cell division, but cytokinesis is actually the physical division of the rest of the cell, the cytoplasm, into two cells. So mitosis, you're going to get your nucleus is going to divide, and cytokinesis is going to take those nuclei and put them into separate cells and divide that cytoplasm into two cells. So I mentioned the checkpoints. There are two checkpoints, G1, which talks about growth, and G2, which talks about DNA synthesis and preparation for cell division. If you fail those checkpoints, you're not going on. Okay, so here is your PMAT, prophase, metaphase, anaphase, telophase. So in prophase, the chromatin is going to condense to chromosomes. So now you're getting the sand material that is condensing to the shape of chromosomes that we know, the long, two double-stranded species, and it's held together by a centromere. Okay, in late prophase, the nuclear envelope begins to break up. Okay, PMAT, so we're in M. M is for metaphase. This is when the chromosomes meet at the center of the cell, midway. It's called the metaphase plate. So they line up and they meet. You can see here that the chromosomes are all in the center. They're all have two strands to them. Okay, so each strand is a duplication. Remember, it's undergone DNA synthesis already, so it's got two strands that are identical. PMAT, okay, PMA, all right, A for anaphase. This is when the chromosomes move apart. You can see that they're getting pulled. So the centriole is attached. It's got a spindle that's attached to the chromosome. It pulls it apart. Now these are identical strands here because we've already undergone DNA synthesis. So we are preparing to divide where we don't lose genetic information. So for a small period of time, we had double the genetic information. It's referred to as 2N, or diploid. Okay, PMAT. All right, telophase is the last phase. This is when everything kind of redoes itself. So the nuclear membrane reforms around the chromosomes. The nucleoli repair, or they reappear, and the spindle disappears. And then you're ready for cytokinesis, where the actual cell pinches off. You get your cleavage furrow, which is the center of the division of the cells, cleavage, and then you're going to get your two daughter cells pinch apart. Okay, protein synthesis. What we have, and we're not showing this, but protein synthesis is when you go from a gene, which is on the chromosome, you go from a gene to RNA to protein. Okay? It's called your central dogma. So the gene is a section of DNA in your chromosome that codes for a protein, usually. There are exons and there are introns. So the exons are what code for your protein. Those have your genes. And then the introns are in between the exons. And these have regulatory components. They attract DNA polymerases and all sorts of different regulatory species. So the DNA is read, and it has C's, A's, T's, and G's. We can go through those. Where the RNA is created, this is inside the nucleus. RNA uses uracil instead of thymine, which we can maybe get into. It's a little bit of a detail. But there are three types of RNA. There's messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA, this is going to be part of transcription. So you have your gene is transcribed. Transcription is like to write something down. And so you take the DNA, you write it down in the language of RNA, which uses uracil. You've also got ribosomal RNA, and as I mentioned, these have a little pocket which can make a peptide bond. Very, very special. And then transfer RNA. These are like the traffic cops that take the amino acids that are to be added to make a protein and bring them where they should be. Now, when you have your RNA and you're going to go to protein, it's called translation because you're translating it into the new language of proteins. So transcription is DNA to RNA. Translation is RNA to protein. And here we've got this central dogma. We've got a double-stranded helix going to a single-stranded RNA. RNA is single-stranded. And then this RNA is processed a little bit. It goes out the nuclear pore, binds onto the ribosome, which is RNA also, and then forms peptide bonds using the transfer RNA. So I mentioned DNA polymerase is the enzyme for synthesizing DNA. Well, you have RNA polymerase, which is the enzyme that synthesizes the RNA. And specifically, you can say messenger RNA. There are different parts of transcription. We're not going to get into this. Okay, so here's your genetic code. And what we have here is usually we have C's, A's, T's, and G's. Cytosine, adenine, thymine, and guanine. That's in DNA. But in RNA, we don't have thymine. We have uracil. So we have G's, U's, A's, and C's. So we have guanine, uracil, adenine, cytosine. So the RNA is read 3 bases at a time. So you have G's, U's, A's, and C's. Here they are here, G, A, C, U. You read that triplet codon, first letter, second letter, third letter. Let's say it's C-U-G. C-U-G. That would be U-U-G. That would be leucine. Leucine is the amino acid that is going to pull to make this polypeptide. It always starts, the polypeptide always starts with AUG. This is methionine. This is the start. And it stops with one of 3 signals, UAA, UAG, or UGA. That's the end of your polypeptide. So first base, second base, third base. That's how you read this. There are 20 different amino acids. Okay, so the tRNA is what's responsible for finding a specific amino acid. Okay, so ribosomes form these arrays where they're all making polypeptides. They're real busy creatures. And then we talked about termination, one of the three codes, UGA, UAA, or UAG. So that's going to be at the end of your at the end of your mRNA strand that is going to initiate the end of translation. Okay. Okay, so this is the information transfer. So just briefly, you've got your double-stranded DNA. It unwinds. It's read by RNA polymerase, which produces the mRNA. Actually, this is DNA. I know it's DNA because it's got thymine in it. It doesn't have uracil. So this is one strand of DNA showing the codons, 3 at a time. AUG, that was your start, your methionine, and then UGA is your stop. And so this is going to produce 8 amino acids long, because 9 is just a stop signal. So your polypeptide will be 8 amino acids. And here we got methionine, proline, serine, leucine, lysine, glycine, arginine, phenylalanine. Okay, and it's all mediated by the tRNAs, which bring the amino acids to the codon. Okay, great. That wasn't that bad. I skipped some parts, and you should too, because you want to concentrate on the cell organelles and their function, and then concentrate on transcription and translation. All those little details about elongation and this and that, not necessarily important. This is a very long chapter, and I don't want you getting overwhelmed. All right, see you later. Bye.